Okay, welcome everybody. This is our first chapter in the genetics text, which is going to serve as an introduction to this particular discipline. Okay, and what we wanted to do is just kind of give you an overview of genetics and its significance in a variety of different aspects of research and policy um, and science. Okay, so we can start with the example of albinism, a trait which involves a lack of pigmentation due to a reduction in the production of a pigment called melanin. Okay, melanin is a brownish black pigment designed to protect DNA, which is the genetic material in all cells, from damage by the UV rays of the sun. And if you inherit an albinism gene from both your mother and your father, you will have this trait. Okay, you're going to be more at risk for conditions such as skin cancer. You'll have easy blistering. All right, the uh, the irises of your eyes will be pink. Okay, you basically have white hair, no pigment. Okay, this is a picture from a Hopi Indian tribe. It was in 1900s that Alej Hrdeka, who was an anthropologist working in the American Museum of Natural History, visited the Hopi in the Black Mesa and reported that among the Hopis were 11 white people who were not Caucasian but Hopi natives, and they had a genetic condition known as albinism, as shown here. Okay? Um, the genetic basis of albinism was first described by Archibald Garrod, who recognized in 1908 that the condition was inherited as an autosomal recessive trait, which means that, again, you have to have two bad copies of the gene in order to show the phenotype, the physical manifestation of this condition. Albinism is caused by mutation in any one of several different genes controlling the synthesis and the storage of melanin. And so this is a biochemical pathway that can have a defect in several places along the production of this brown pigment. So there's different types of mutations that can occur at each gene responsible for each step. Any one can lead to albinism. The form of albinism among the Hopis is likely the oculocutaneous albinism type 2, which is a defect in a gene called OCA2 on chromosome number 15. Remember that from your basic biology that humans have 23 pairs of chromosomes, and these are numbered in order of size, with 1 being the largest and 22 being the smallest autosome. Those are chromosomes that contain body information. Now, the 23rd pair are your sex chromosomes. And so chromosome 15 would be relatively small, but still containing critical information. The Hopis aren't unique in having people with albinism. It's found in almost every ethnic group and is described in ancient writings, and it's got a frequency of about 1 in 20,000. In the Black Mesa, it was about 1 in 200, which is 100 times higher than other populations. So that's a little unusual, right? Why so high? Why is this condition so frequent among the Hopis? And the answer to this question isn't completely known, but individuals who studied albinism in the Hopis think that the high frequency of the albino mutation is due to the special place that albinism occupied in the Hopi culture. For much of their history, they considered members of their tribe with albinism to be important and special, and people with albinism were considered attractive, clean, and intelligent. So having a number of people with albinism in one's village was a good sign, a symbol for the people of the village that they had pure Hopi blood. Members of the tribe with albinism performed in Hopi ceremonies and held positions of leadership, often as chiefs and healers and religious leaders. So this is one explanation for why this is so prevalent, right? It's a cultural explanation. These individuals were given special treatment in everyday activities. The Hopis have farmed small garden plots at the foot of the Black Mesa for many years. Every day throughout the growing season, the men of the tribe trekked to the base of the Black Mesa and spent much of the day in the southwestern sunlight, tending to their corn and vegetables. With little or no melanin in their skin, we have increased incidence of skin cancer, which we would expect. As a result, we don't see well in the bright sunlight due to the lack of melanin as well. The male Hopis with albinism were then excused from farming and allowed to remain behind in the village with women of the tribe performing other duties. Throughout the growing season, the men with albinism were the only male members of the tribe in the village with the women during the day, and so they had a mating advantage, and this increased the frequency of this particular gene, which we would call an allele, in the population. Okay? Now, another thing to consider, all right, is that generally speaking, in the Hopi tribes, the breeding patterns were such that Hopis would breed with other Hopis and not outbreed, meaning intermarry with individuals outside their tribe or their race. So what that results in is in a population that was founded by a specific number of individuals, in the event that that gene was originally present in the population in a higher percentage than it is in the general population, if that population continues to breed amongst themselves, the frequency of that gene will be higher than other populations. Okay? And that's called founder effect. Okay? We're going to talk about the importance and the history of genetics and some fundamental terms and principles of genetics as well. Genes influence our lives. One of the things that we have to understand, all right, is that remember that genetic information has both a physical and a behavioral component in many cases, right? Any trait, right, that is something that we can measure, physical manifestation, is going to have both a genetic and an environmental component. Okay, the genetic component is that part that is inherited, and the environmental component is what that physically looks like when it interacts with the environment. Okay, we could take an example like body weight. Okay, when you're born, there's a certain cluster of genes that are going to set the window for your upper and lower body weight. Okay, if you're above the ceiling or below the floor, you have a condition known as disease. And if you're way above the ceiling or below the floor, then you're going to die. Okay, where you lie within that window set by the genetic component is determined by your interaction with the environment, how much you exercise, how much you eat. Okay, so it's the combination of those two things that produces the trait. How much the environment matters and how much the genetic composition matters is something that can be estimated by studying individuals who are genetically identical, such as identical twins. Okay, and for every trait, the biological and the environmental component are going to be a little bit different. Okay, genes can contribute to our personality. Okay, there are genetic traits that have been described in psychology, such as obsessive compulsive disorder and schizophrenia, that have been linked to certain clusters of genes inherited at fertilization. Okay, and again, with these, there is also an environmental component. Okay, as a result, genes are fundamental to who and what we are. Okay, these proteins, which are what the gene products are basically, are going to contribute to characteristics of cells and tissues and that's going to contribute to characteristics of the organism. Now, why would this influence things like personality? Well, you have to remember that in the systems in the body, such as the nervous system, that's a tissue, right? Your brain, your brainstem, your spinal cord are made of living tissue. And the way that those tissues operate is going to be determined by their genetic content, okay? So this is going to affect the way that the brain operates, the way that the brain perceives stimuli, the way that the brain analyzes the stimuli and produces commands, okay? All of this is going to be, in part, the result of our genetics, okay? It also is going to have a significant amount to do with our interaction with the world around us, okay? Although the science of genetics is relatively new compared with things like astronomy and chemistry, people have understood the hereditary nature of traits and practiced genetics for thousands of years, okay? We began this all the way back when we were doing animal husbandry and agriculture, okay? Originally, when humans appeared on the planet, all right, what they ate was what they killed or what they found out in the wild, okay? But we eventually figured out that we could select the seed from plants that produced things that we like to eat and plant those seeds, and then the next crop that would appear would have more of whatever it is we were pursuing in order to augment our diet, okay? And that's an example of selection, okay, based on specific traits. And the same is true with animal husbandry, right? We had animals that could provide food for the group, and what we would do is breed them for things like milk and egg production, okay, and the ability to assist us with things like, let's say, important operations in a farm or a ranch setting, okay, such as the domestication of the horse, right? Would just be one example, right? So this has been going on since man has been around, okay? Today, the major crops and animals used in agriculture are different from their wild progenitors and have undergone extensive genetic alteration to improve their yields and desirable traits, okay? The Green Revolution, which expanded food production in the world in the 50s and 60s, relied both on the primitive methods of selection, but also the application of molecular biology in order to improve certain crops, provide particular yields, and even add, for instance, nutritional components to some of the fruits and vegetables. Today, genetically engineered corn, soybeans, and other crops are a significant proportion of the world's food supply, okay? And you can see here in this figure, all right, an example of genes that contribute to disorders, right? This is an example here in figure A, an x-ray of the hand of a person suffering from diastrophic dysplasia. That's on the green panel in the bottom. It's a hereditary growth disorder that results in curved bones and short limbs and hand deformity. And we compare this with an x-ray of a normal hand on the top. Note in figure B, right, that diastrophic dysplasia is due to a defect in the SLC26A2 gene on chromosome 5. So it's a pretty large chromosome. This is an example of a structural disorder, okay? This likely is an autosomal dominant, okay? Generally, when you have diseases that affect structural components in the body, they're inherited as dominant alleles, meaning you only need one copy of the defective gene for the trait to show up. Whereas in diseases that are the result of the lack of something like a specific enzyme, these tend to be recessive disorders. Okay, and the reason for that is in the example of a defective enzyme, such as in the albinism case, you need two bad copies of the gene to completely nuke the specific enzyme responsible for the melanin production. Okay, whereas in a structural disorder, if half of the components are defective, then the defective characteristic is going to show up in the organism. Okay, it's kind of like building a building a building where half the bricks are bad, right? Genes are critical in agriculture. Okay, you can see here genetic techniques developed to generate new high-yielding strains of crops. Okay, one of the leaders of this, pictured on the left, was Norman Borlaug, a leader of the development of new varieties of wheat that was part of the Green Revolution. He got the Nobel Prize in 1970, and you can see the comparison of high-yielding and low-yielding rice, right? This is traditional rice. This is high-yield rice, okay? We have the ability, with a variety of techniques, to take a gene of choice and place it in the DNA of a particular plant, generate tissue culture of that plant, grow it up, gather seed, and then generate now this this modified version of the wild organism that has a desired trait in it. Okay. The pharmaceutical industry is another place where genetics plays an important role. A lot of drugs and food additives are synthesized by fungi and bacteria, which have been manipulated to make them efficient producers of substances. The biotech industry employs molecular genetics to develop and mass-produce substances of commercial value. Anti-malarial drugs, growth hormone, insulin, clotting factor, and antivirals, as well as enzymes, antibiotics, vaccines, and other compounds, are produced commercially by genetically engineered bacteria and other organisms. Genetics has also been used to generate bacterial strains that remove minerals from ore, break down toxic chemicals, and produce biofuel. Okay, so the applications are vast. Okay, genetics also plays a critical role in medicine. Physicians recognize that a lot of diseases and disorders have a hereditary component to them, including rare disorders such as sickle cell anemia and Huntington's, and common diseases like asthma, diabetes, and hypertension. Advances in the field have resulted in important insights into the nature of disease, such as cancer, and the development of diagnostic tests, including those to identify disease-causing mutations and pathogens. Genomic data are helping to usher in a new era of personalized medicine, and the development of low-cost sequencing methods allows us to get a person's complete genome and sequence and give us information about particular mutations in their genome that might provide inroads for particular treatments. And then there's the possibility of gene therapy, where we alter the gene that is defective in producing the disease in order to remedy the disease. Okay, and we have ways to do that now that are actually based on some bacterial methods of recombination. Okay? Even though an understanding of genetics is important to everyone, it's critical for somebody studying biology, like y'all are. Genetics provides one of biology's important principles. All organisms use genetic systems and have a lot of features in common. Genetics undergirds the study of a lot of other biological disciplines. Evolution, for example, is the genetic change taking place over time, and while we can't directly see it in real time, we can observe natural selection as quickly as one 24-hour period. We can see that, for instance, on a petri dish using drugs to select for resistant strains of bacteria in the face of a particular antibiotic in the media. Okay. And that's the mutations along with the selection are the engine that drives evolution. Okay? Developmental biology relies on genetics. Tissues and organs develop through regulated gene expression, as shown here. Okay? This is an example of a fruit fly embryo showing localized expression of the engrailed gene, and that helps to determine the body segments in the adult fly. Segmentation is an important evolutionary advancement in body plan because it allows for a more complex organism that is more easily adaptable to its surroundings. Okay? Even fields like taxonomy, ecology, and animal behavior are making an increasing use of genetic method. The study of almost any field of biology or medicine is incomplete without an understanding of these methods. Okay? Now, when we talk about a genome, right, we're talking about the complete set of genetic instructions for a particular organism. So this is basically the nucleotide sequence from the first to the last chromosome in complete detail. Okay? The alphabet of the instructions that are going to shape the way that the cells and the tissues and the organs work. Okay? Genetic material can be either RNA or DNA. Now, RNA can be used as a genetic material for entities known as viruses, all right? While DNA is the genetic material that is going to drive the expression of proteins in both prokaryotic and eukaryotic cells, okay? The RNA in prokaryotic and eukaryotic cells acts as a go-between between the information in the DNA and the production of the protein. Okay? So the information flows from the DNA to the RNA to the protein in living organisms. All right? In viruses, it's a little different. Okay? In viruses, you can have either RNA or DNA, and the RNA or the DNA can either be single or double-stranded. Okay? But you have to understand that viruses aren't technically living things because they can't reproduce without a host. Okay? The genome is copied during replication. Now, the reason this is possible is because DNA is double-stranded. All right? There's a strand of DNA that runs in one particular direction. We call the 5' end of that strand the beginning, and the 3' end the end. And then there's a second strand right next to it that runs in the opposite direction. Okay? So it runs from 5' to 3', but it's lined up so that its 5' end is next to the 3' end of the other strand. And this is essentially a photocopying mechanism for DNA, because during the process of DNA replication, where we make new DNA for new cells, those two strands will disassociate from each other and will be used to guide the production of new strands of DNA. And there are rules that dictate what goes into those new strands. The rules of complementarity dictate that whenever there is a building block in the DNA polymer called an adenine, that there will be a thymine incorporated into the complementary strand, and whenever there is a DNA block called a guanine, a cytosine, will be incorporated into the complementary strand. Okay? So the idea here is that since you have a template to guide the production of the new strand, that when you're done with the whole process, you have a complete copy of the information in the original cell that's given to each new cell upon the completion of cell division known as mitosis. All right? And the only place that breaks down, all right, is during a mutation, which is a mistake in the reproduction of DNA that results in an incorrect information block being incorporated into that DNA. Okay, and if that isn't corrected, because there are proteins, enzymes that take care of goofs in the process and normally um, are able to fix the damage before it becomes part of the new cell, right? But if that isn't caught, that's a mutation, and a mutation can have three possible outcomes. It can be beneficial to the organism, meaning it's more likely to survive. It can be bad for the organism, meaning it's less likely to survive, or it may not have any effect. Okay? But it's mutation that is part of the driving force of evolution. Okay? The coding system for genetic information turns out to be the same in all living organisms. Okay? So when we look at a living cell, we always see the instructions as DNA, okay, double-stranded DNA. Or organisms are thought, as a result, to have originated from a single living common cell approximately 4 billion years ago. Now, what we don't understand, okay, is where that living cell came from. All right, how do we get from non-life to life? Okay, and there are various theories. Okay, one theory is that that event, whatever it was, happened here on planet Earth and led to life. Another possibility is that something living came here from outer space. All right, we don't know, since we weren't here at the time, what the answer is. Okay, all we know is that once we had the first living cell, that what derived from that owes many of its characteristics to that original living cell. Okay? Okay. Each DNA molecule is very small, but because all cells contain genetic information, there's a lot of DNA in the world, and scientists estimate that the total DNA in the biosphere is about 5.3 times 10 to the 31 megabase pairs. A mega is a million, okay? And that weighs some 50 billion tons. Storing information content in the world's DNA would require 10 to the 21st computers, each with the average storage capacity of the world's four most powerful supercomputers. Scientists are now cataloging and measuring the world's biodiversity by analyzing DNA. As an example, researchers aboard the ship Terra surveyed the world's oceans for organisms by isolating DNA from seawater during a 3.5-year voyage, and they collected 35,000 samples and extracted DNA from them. It was then sequenced and analyzed and revealed the presence of 150,000 genetically distinct types of eukaryotes. Okay, remember that a eukaryote is either a single or multicellular organism that has an organization consisting of membranous and non-membranous organelles and a true nucleus, meaning a membrane-bound nucleus, which is where the DNA is located. The DNA in the nucleus is combined with proteins that compact and protect the information in the DNA, and that complex of protein and DNA is called chromatin. Okay? Most of these eukaryotes were newly discovered single-celled organisms. They also detected about 5,000 viruses, only 39 of which were previously known. There are different divisions of genetics: transmission, molecular, and population genetics. Okay? The study of genetics consists of these different subdisciplines. Transmission genetics encompasses the basic principles of heredity and how traits are passed from one generation to the next. This subdiscipline addresses the relation between chromosomes and heredity, and the arrangement of genes on chromosomes and mapping. Here, the focus is on the individual organism and how an individual inherits its genetic makeup and how it passes its genes to the next generation. Okay? Molecular genetics is basically the manipulation of the genome in order to either analyze or produce novel traits. Okay, so what happens in molecular genetics is that we're able to take DNA from different sources and paste it together using enzymes such as restriction endonucleases and ligases, and generate constructs or put constructs in pre-existing nuclear DNA and change the characteristics of that organism. Okay? So what could some applications be? We might want to produce large amounts of a particular drug. We might want to analyze whether or not a particular change in a gene mimics a disease that we see in a particular organism, like a human. Okay? We might want to take a working gene and basically obliterate its function to see what the effect is on that organism's development and behavior. Okay, so there's any number of applications of molecular genetics, and you can perform molecular genetics on eukaryotic organisms, prokaryotic organisms, and they can be for a variety of end applications. This is one of the reasons that developing skills as a molecular biologist means that you can serve in a variety of different laboratories, investigating a variety of different systems, because you have the ability with these tools to generate these constructs, analyze them, and then attach to them particular characteristics. Okay? Population genetics is the study of genetic composition of populations and how their gene pools change over time. The study of genetics in these three subdisciplines is convenient and traditional, but we should recognize not only that they overlap, but also that each one can be further divided into more specialized fields, such as chromosomal genetics, biochemical genetics, quantitative genetics, and so on. By the same token, genetic studies can be subdivided by organism, right? Fruit fly, corn, yeast, and each of these organisms can be studied at the level of transmission, molecular, or population genetics. Okay? Modern genetics is an extremely broad field and encompasses a lot of interrelated disciplines. Okay? What we want to look at now are some model genetic organisms. Okay? Now, one of the things to appreciate about a model genetic organism is that in a lab setting, it has to have a short generation time. It has to have a genome that is relatively well characterized, and it has to be inexpensive to maintain, which usually means the organism is rather small. Okay, the simpler the genome, the easier it is to work with, right? Because it's easier for you to determine what the changes that were produced or the changes that you found had as an effect on that particular organism, right? So this is why when we talk about model organisms, you'll be seeing a lot of laboratories working with bacteria or with yeast, or in plants, you might see a lot of labs working with Arabidopsis, okay? You might see a lot of labs working with insects such as Drosophila, okay? So there's a variety of reasons for that, but what all of those organisms have in common is they reproduce pretty quick, okay? Their genomes are well known, and they're cheap, okay? So that's what we're going for here when we talk about these model organisms. Okay? Model genetic organisms have characteristics that make them useful for this analysis, and there's 6 that have been intensively studied genetically. Okay? And these, again, are the characteristics that we like, right? The short generation time, the ability to carry out control crosses, produce a lot of progeny, right? So you have big numbers in order to determine what the effects of a change is. Laboratory friendly, meaning inexpensive to maintain. A lot of genetic variants that are either already out there or the ability to produce a lot of genetic variants, and a lot of knowledge about how their genetic systems work. Okay? So all of this makes a perfect model organism. This is why you're not going to see labs working with like transgenic elephants. Okay, too expensive, too large, generation time too slow. All right. So what are some examples of model genetic organisms? Well, we can take the fruit fly. Okay. So this is a picture of the fruit fly. This is Drosophila melanogaster. Okay. It is an insect. The benefits of utilizing Drosophila, okay, short generation time, inexpensive to maintain, okay, and a lot of characteristics that are well known, and a genome that we know basically front to back, and the ability, as a result of specific strains, to move pieces of DNA around inside the organism through the use of mechanisms such as transposable elements. Okay? What are some characteristics that can be mapped on the fruit fly? The eye color, the wing shape, the body color, okay? The behavior in some cases. Okay? It's also a eukaryote, which means that the production of genes that have been manipulated is very similar to the way that those genes would be expressed and produced in something like a human. Okay, so the closer you get to the organism that you're really gunning for, the more likely that the results from your model are going to be meaningful. Okay, another example, E. coli. Okay, E. coli, small, inexpensive, rapid generation time. Under optimal conditions, once every 20 minutes they divide. Okay, can be easily genetically engineered. Okay, bacteria such as E. coli can either reproduce asexually, or in some cases they can have sexual transmission of genetic material through a process called conjugation. Okay, bacterial genetics is unique because in bacterial genetics, the process known as transcription, which is the production of RNA directly from DNA, and translation, which is the production of protein from RNA, happen in the same location and at the same time. Okay, and you can also use mobile genetic elements to affect the characteristics of an E. coli. Right, plasmids are an example of a genetic construct that can be transferred from bacteria to bacteria through processes like conjugation. You can introduce plasmids through a process called transformation, and thus provide new characteristics for those particular cells. And you can also use it as a way to manufacture large quantities of a construct that you've built in the test tube. Okay, you can, for instance, produce a plasmid by splicing together different pieces of DNA and then use the bacteria to make large amounts of that genetic material for another downstream application. Okay? Keep in mind, though, that this is a prokaryote. All right, so a lot of the processes of gene expression that occur in a eukaryotic cell do not occur in a prokaryote. Okay, this is C. elegans. All right. C. elegans, again, actually feeds off of E. coli, all right, and has a relatively small genome. It is a nematode, right, so it's a worm, a soil-dwelling nematode, and it has a relatively simple nervous system and a genome that is well characterized and a relatively short generation time. So what we can do with C. elegans, and it is a eukaryote, is that we can modify genes or study pre-existing mutations that affect the development and the behavior of the organism and have some idea what the effect might be in another eukaryote, okay, such as a human. Right? Arabidopsis thaliana, okay, is the thale cress plant. It has an extremely short generation time, It can be genetically manipulated either in tissue culture or using techniques such as Agrobacterium tumefaciens, right, in order to introduce um, novel gene constructs that can modify the behavior of the organism, either for the purposes of studying that particular gene or producing a condition that you want to mimic in some other target crop species, okay, such as something like wheat or corn. And then we get to the mouse, Mus musculus, okay. This is commonly used to mimic human disease models, all right. One of the things we can do with a mouse is that we can harvest the eggs from a female mouse, and we can introduce novel genetic material from a different source, integrate it site specifically into the location of a particular gene, and affect the characteristics of the organism that results when that egg is fertilized. Okay, we can also do this um, at the blastocyst level. Okay, which is a little bit after the egg and the sperm have met and we've affected fertilization. Okay, and then we have baker's yeast, Saccharomyces cerevisiae. This is the smallest eukaryote that we work with in the lab generally. Okay, and the advantage of this, of course, is that it is a eukaryote. It's a fungus, but the processing of particular genes in Saccharomyces is going to be very similar to what we see in larger eukaryotes. So we can take a lot of the information that we get from Saccharomyces and apply it to things like humans. Okay, short generation time, well-known genome. It engages in both asexual and sexual reproduction. Okay, and it also, along with E. coli, is frequently used in the production of large amounts of material for the purposes of the production of food, the production of medicine, the production of any number of industrial products. Okay, so we can use the yeast and the bacteria as little factories in order to generate large amounts of something that we're after, because they reproduce so quickly under optimal conditions, and that means that they will express whatever they have in them, in the nucleus, in relatively large quantities, which are easily harvested. Okay? Interesting here, this is the zebrafish. This is another model organism. Okay, it is a eukaryote. All right. The model genetic organism, illustrated by the use of the zebrafish. An example here was to identify genes that affect skin pigmentation in humans. For a long time, geneticists recognized that differences in pigmentation among human ethnic groups were inherited, but the genes causing these differences were largely unknown. The zebrafish has become an important model in these studies because it is a small vertebrate that produces a lot of offspring and is easy to grow in the laboratory. The mutant zebrafish, called golden, has light pigmentation due to the presence of fewer, smaller, and less dense pigment-containing structures known as melanosomes. Okay? So scientists hypothesized that the light skin in humans might result from a mutation similar to the gold mutant in zebrafish. Taking advantage of the ease with which zebrafish can be manipulated in the lab, they isolated and sequenced the gene responsible for the gold mutation and found that it encodes a protein that takes part in calcium uptake by melanosomes. They then searched a database of known human genes and found a similar gene called SLC24A5 that encodes a protein that has the same function as human cells. They examined the human population and they found that light-skinned Europeans possess one form of this gene, and darker-skinned Africans, Asians, and Native Americans have a different form. Many other genes also affect pigmentation in humans, as illustrated by mutations in the OCA2 gene that produces albinism in the Hopis that we talked about earlier. Nevertheless, the SLC24A5 appears to be responsible for about 40% of the difference in pigmentation between Africans and Europeans. So this illustrates the power of a genetic model. Okay, so what they're doing here is they're looking at pre-existing mutants and they're comparing that to conditions in a different organism, and they're hypothesizing that the effect in one could be responsible for the effect in the other. And so what you do is you check out that information by looking at the changes in the mutant versus the changes in the human DNA. Okay, and that can steer you towards what actually might be responsible for those changes. Okay, we noted earlier that we've been using genetics for thousands of years. Okay, we talked about the understanding of heredity. Evidence that people understood and used heredity in early times was found in the development of farming and animal husbandry, okay, as we discussed early in the talk. Ancient Jewish writings indicated that they understood the inheritance of hemophilia, right? Other crops and domesticated animals were developed by cultures in Asia, Africa, and the Americas during the same period, approximately 4,000 years ago. Ancient writings demonstrate that early humans were aware of their heredity. Hindu writings dating 2,000 years ago suggest that a lot of traits are inherited from the father and that differences between siblings are produced by the mother. The Talmud, which is a Jewish book of religious laws based on oral traditions dating back thousands of years, presents an uncannily accurate understanding of hemophilia. It directs that if a woman bears two sons who die of bleeding circumcision, any additional sons that she bears should not be circumcised, nor should their sons or their sisters be circumcised. And that advice corresponds to the X-linked pattern of inheritance of hemophilia that exists today. Some early concepts of heredity were incorrect, but reflect human interest in heredity and our attempts to explain inheritance and traits. You can see here some examples, right? Genetic techniques to produce food crops, all originating from its ancestor, grass, right? Ancient people were aware of this, right? Some of these early theories about inheritance, okay? The ancient Greeks gave careful consideration. Greek philosophers developed the concept of pangenesis, in which specific particles called gemmules carry information from different parts of the body to the reproductive organs, which are then passed to the embryo at the moment of conception. Even though incorrect, the concept of pangenesis was influential and persisted into the 1800s. Okay? The concept of pangenesis led the ancient Greeks to propose the notion of inheritance of acquired characteristics, according to which traits acquired during a person's lifetime become incorporated into their genetic material, and they pass those on to their offspring. Lamarck was a proponent of this idea. He was doing his work during the mid-1700s to the mid-1800s. Okay? He was a proponent of the idea incorporated into the theory of biological change. The notion of inheritance of acquired characteristics is no longer accepted, but it remained popular. Okay? Additional developments in our understanding of heredity occurred during the 17th century. Dutch eyeglass makers began to put together simple microscopes in the late 1500s, and that enabled Hooke to discover cells in 1665. Microscopes provided naturalists with a new and exciting view of life. Perhaps it was excessive enthusiasm for this new world of very small organisms and structures that gave rise to the idea of preformationism, which was the idea that inside the egg or the sperm there was a fully formed miniature adult called a homunculus, which simply enlarges in the course of development. Preformationism meant that all traits were inherited from only one parent, from the father if the homunculus was in the sperm, or from the mother if it was in the egg. Although many observations suggested that an offspring had a mix of traits from both parents, this remained popular for many centuries. Okay? Another notion of heredity was blending inheritance, which proposed that the traits of offspring were a blend or a mix of parental traits. This idea suggested that the genetic material itself blends much as blue and yellow pigments blend to make green paint. It also suggested that after having been blended, genetic differences couldn't be separated in future generations, just as green paint can't be split back out into blue and yellow pigment. Some traits do appear to exhibit blending, but we realize today that the genes themselves don't blend, although the phenotype they produce, in some cases, can be a blend. Okay? So here you can see, again, pangenesis versus what we know today as the germplasm theory. Right? In the germplasm theory, which was basically a conglomeration of efforts by Darwin and Weismann, the idea was that the cells in the reproductive organs carry a complete set of genetic information. Okay, August Weismann was one of the people that laid to rest the notion of inheritance of acquired characteristics. So he was able to go directly against Lamarck. He did this by cutting off the tails of mice for 22 consecutive generations and showed that the tail length in the descendants remained long. Weismann, as a result, proffered the germplasm theory that we've discussed already. When we look at the change in the science of genetics over time, we can begin with Mendel. His pivotal 1866 publication on experiments with peas revealed principles of heredity basically rediscovered as considered in more detail in material we'll hit in Chapter 3. Once the significance of his conclusions was recognized, other biologists began to conduct similar genetic studies on mice, chickens, and other organisms, and the results showed that many traits indeed follow Mendel's rules. In 1902, the acceptance of Mendel's theory of heredity led to work by Walter Sutton, who proposed that genes, which are the units of inheritance, are located on chromosomes. Thomas Hunt Morgan discovered the first mutant fruit fly in 1910, and he used them to unravel details of transmission genetics. Fisher and Haldane and Wright laid the foundation for population genetics in the 1930s by integrating Mendel's genetics with evolutionary theory. Geneticists began to work with bacteria and viruses in the 40s. The rapid reproduction and simple genetic systems that these organisms had allowed study of the organizational structure of their genes. About this same time, evidence accumulated that DNA was the repository of genetic information. Watson and Crick, along with Wilkins and Franklin, described the three-dimensional structure of DNA in 1953, ushering in the era of molecular genetics. We should note also that it was Schleiden and Schwann, that came up with the cell theory. Now, this was an outgrowth of work from Leeuwenhoek, who invented the microscope and noted that all living things were made of cells. Schleiden and Schwann, utilizing the microscope, fleshed out the cell theory, which stated that all living things are made of cells and all cells come from pre-existing cells. Okay? And at the time, that was pretty revolutionary. Okay? Now, one of the things that's happened in recent years is that we've taken some techniques that were developed in the 50s and 60s and the 70s And we've been able to modify them so that we can get a detailed snapshot of the genetic information in every organism from front to back. Um, One of these techniques was called Sanger dideoxy sequencing, and we'll get more into detail about exactly how this works when we get into more molecular biology techniques. But using this technique and using an application of this technique in robotics, we've been able to sequence the entire human genome from front to back. And so we know in a representative organism the base sequence of every single chromosome. Now, that's a lot of information, and what we're in the process of doing now is attempting to attach function to these pieces of information. Okay, a lot of what came out of this sequencing effort was the location of known genes with known functions. Okay, but that was a small fraction of the total information, and so what we've done is we've been looking at different regions of the DNA produced by the Human Genome Project and looking for naturally occurring changes and trying to attach particular conditions to those changes so that we can more fully understand mutation in human disease. Okay, this is assisted by the use of bioinformatics, where we're able to assign or we're able to, as a result of generating a large database of both protein and gene data, the ability to, across organisms and within organisms, derive functions by comparison, followed by experimentation. Okay, and this database is getting bigger every day, in addition to the fact that the ability to sequence large amounts of DNA and great distances of DNA has improved several orders of magnitude in just the past decade. Okay? Some fundamental concepts in genetics critical to the rest of this course. Cells are of two basic types, eukaryotic and prokaryotic. Structurally, cells consist of two types, although evolutionarily the story is a little more complex, and we'll get into that in Chapter 2. Prokaryotic cells lack a nuclear membrane and don't generally possess membrane-bound organelles, and eukaryotic cells are more complex, having a membrane-bound nucleus and membrane-bound organelles, and features such as chloroplasts and mitochondria. The gene is the fundamental unit of heredity. The precise way in which a gene is defined varies depending on the context. At the simplest level, we can think of a gene as a unit of information that encodes a genetic characteristic. We'll expand on this definition as we learn more about what genes actually do. Genes come in different forms called alleles. A gene that specifies a characteristic may exist in different forms. These forms are called alleles. An example, a gene for coat color in cats can exist as an allele that encodes for black fur or as an allele that encodes for orange fur. Genes confer phenotypes. An important concept in genetics is the distinction between traits and genes. Traits are not inherited directly, rather genes are inherited, and genes, along with environment, determine the expression of the trait. Genetic information that an individual organism has is its genotype, while the trait is its phenotype. As an example, the albinism seen in the Hopis is a phenotype, and the information in the OCA2 gene causes albinism in the genotype. And finally, genetic information is carried on both DNA and RNA. Okay? Genetic information is encoded by the structure of nucleic acids, which comes either as DNA or as RNA. Nucleic acids are polymers of repeating units of nucleotides, which have a sugar, a phosphate, and a nitrogenous base. The nitrogenous base in the DNA can either be an adenine, cytosine, guanine, or thymine. The sequence of these bases contains the genetic information in the polynucleotide. DNA consists of two complementary nucleotide strands. Most organisms carry their information on DNA, but some viruses have RNA as their genetic information. The nitrogenous bases of RNA are adenine, cytosine, guanine, and uracil. Genes are located on chromosomes. Okay? The vehicles of genetic information within a cell are chromosomes. These are DNA molecules and associated proteins. That material is called chromatin. The cells of each species have a characteristic number of chromosomes. As an example, bacterial cells normally have a single chromosome. Human cells have 46 and pigeons have 80. Each chromosome has a large number of genes. Chromosomes separate through the process of mitosis and meiosis. The process of mitosis and meiosis ensure that a complete set of an organism's chromosomes exists in each cell that results from cell division. Mitosis is a separation of chromosomes in the division of somatic cells, while meiosis is the pairing and separation of chromosomes in the division of sex cells to produce gametes, or reproductive cells. Genetic information is transferred from DNA to RNA to protein. This is called the central dogma of biology. Many genes encode characteristics by specifying protein structure. The genetic information is transcribed from DNA into RNA and translated from RNA into protein. And it's the proteins, in large part, that determine the genetic component of the traits. Mutations are permanent changes in genetic information that can be passed from cell to cell or from parent to offspring. Mutations affect the genetic information of a single gene. Chromosome mutations alter the number or the structure of chromosomes and therefore affect many genes. Many traits are impacted by multiple factors. Many genes interact in complex ways with each other and with the environment. Human height, as an example, is affected by many genes as well as environmental factors like nutrition. Okay? And we're going to investigate each of these components in subsequent chapters. Okay? And finally, evolution is genetic change. It can be viewed as a two-step process. First, genetic variation arises, and second, genetic variants increase in frequency, whereas other variants decrease in frequency. Okay? What I want you guys to do is to hit the sampling course and do the homework in preparation for the first exam. All right? Remember that this is an 8-week course, so we're moving pretty quick here. If you have any questions, please get in contact with me during my office hours, which are Friday from 5 to 7. All right, either by email or by phone. You can call my cell, or if you want to do chat, we can do that as well. Okay, I'll see everybody in the next chapter.